Before we get into the genome report for a particular job, I want to discuss the summary section, which is part of that genome report that appears at the top of it. So there are a number of different things that give you some indication of the quality of your genome. It tells you first genome ID, which is the unique numerical identifier for this particular genome. Genomes within the resource may have the same name, but their genome ID will always be unique. Genome name is the next row down. The reference genomes or genome or genomes is a high quality public BV-BRC genome that's close to the one submitted that's used as a reference in the comparison. Coarse consistency is the percent of genes predicted to be in this genome. Fine consistency is the percentage of genes that occur as predicted. Completeness is the percentage of universal genes in the genome. This is a smaller subset of all the genes that are used in evaluation, and we use a specific program called eval-G and another one called eval-Con to do that particular evaluation. Contamination is the percent of the genome DNA that does not belong. So when it's evaluating your genome, it's looking for things that it expects to be there in terms of genes, and it's also identifying things that it doesn't expect to be there. Not only genes that it doesn't expect, but also DNA that it doesn't expect. Evaluation group is a group used to determine the universal genes, and the universal genes are described here as part of the completeness. Contig count, that's easy, number of contigs. DNA size, those are the base pairs present in your genome. Contig N50 and contig L50 are two evaluations to see how good the assembly is in terms of the number of contigs and the size of those contigs, and there are enough of the contigs of sufficient size that we feel comfortable with it. Over-present roles. In BV-BRC, a role is a gene, and if it's an over-present role, it means that there were a number of genes that were found too many times, more than we expected. Under-present roles, those are genes that were found too few times. Did your genome not have things that everybody else has that we expected to be there? Completeness roles, this ties back into those universal roles, that subset. So it's just telling you the number that it considered to be in this genome, the universal roles, which determine the completeness. The total distinct roles are all the genes in the genome. Protein encoding genes with a functional assignment, those are the number of genes that have an actual name that is not hypothetical. We are sort of figuring out what those genes do. If they're called hypothetical, we're not sure, one, if they do anything, and two, if they're even ever annotated. Protein encoding genes without functional assignment, those are the number of hypothetical proteins. Percent protein encoding feature coverage. So it's looking at all the genes and it's doing the percent of the coding regions versus the number expected in a genome of this size. So it's going to say, well, in this genome, we expect it to have this many genes, and then looks at the number that are actually there. Percent features that are hypothetical. The percent of proteins that are annotated is hypothetical. And then percent features that are in local protein families. We've talked before about how the annotation pipeline identifies two classes of protein families if you specify to the genus level in your taxonomy. There's the cross-genus families, which are the global protein families, and there are the genus and species specific to a particular genera. Those are the local protein families. So this is just looking at how many of the proteins in my particular genome are in genus-specific protein families. Okay, I know it's a lot of information, but let's look at what it actually looks like. So here is the genome report for our job. It's giving me the unique identifier. Here's the name I established for it. This is the genome that was used as the reference genome. Were I to click on that number, it would take me to the genome landing page for that genome. Were I to click on this number, it would take me to the genome landing page for my genome. Course consistency looks good. Fine consistency looks good. Completeness looks great. And contamination looks good. It looks like we have everything that's expected to be here. This is the evaluation group, the group of genomes that it was looking at. It was 32 contigs, this particular genome. This is the size of the genome. These are probably things that you would report in the results section of your paper if you were describing this thing. The N50 and L50 to describe the assembly quality. Now, over present rules and under present rules. How many genes did it have that were more than we expected? So that's 3. How many genes did we expect to be there that weren't? So that's 7. And these will all be discussed down below in the potentially problematic rules. Then we have the total number of predicted rules that we thought would be in the genome and the completeness rules, those particular genes that were used to determine the percent completeness. So the total distinct rules are all the genes that were in the genome. Protein encoding genes with a functional assignment, most of them. Generally a bacterial genome, often it's like 30% hypothetical, but this is much better than that. So in this particular Haemophilus influenzae genome, most of the genes have a functional assignment. It's non-hypothetical, and only 327 are hypothetical. The percent protein encoding feature coverage, 99, that's good. Percent of features that are hypothetical, only 17. I think that's pretty great. And then the percent of features that are in local protein families, 97%. There are enough genomes in Haemophilus that when we run the pipelines to determine the protein families, it's pretty complete. This indicates to me that maybe we're near a kind of saturation because most of those are turning up in protein families. So that's a good thing. So this genome is looking good. So let's talk about the potentially problematic rules. Up here, we have the over-present and under-present rules, and there were 10 genes that were problematic, and here they are. Now, some of these, if you look in the comment section, they weren't found in the reference genome, and here they aren't found in the reference genome. So those were things that we didn't expect, and then we expected to be there, but were not. And then we have things that didn't occur, so they're calling them over-present rules. And you can see that they're describing them here that are in our particular genome. And sometimes there's some interesting things that they say, and they also give pointers to the contig and the gene that are there. So this one, we would have expected it to have 1 gene, but it actually had 2. And these are the 2 genes that are there in our annotated genome. And the same situation is here. And sometimes you can use this to try to find pseudogenes and things like this, or probably more accurately, assembly errors, like things that are divided on 2 different contigs. Part of a gene is on each contig. So this might be an area of interest to you, especially if you're interested in particular genes. And let me point this out. Before you report that a gene is missing because of issues that might occur with assembly, you better blast in that sequence about the genome to see if you've got that particular problem. because it could just be that the assembler couldn't join them together for some reason. And then finally, we have the potentially problematic rules by contig. And this is telling you where these things are. And this can be useful if you have something like a novel plasmid that's in your genome. For example, this contig had one problematic feature out of 288. So I wouldn't think of plasmid for this. But if I had a contig that I had things that weren't expected in this genome, let's say 20 genes that had been identified as potentially problematic, and they all occurred on the same contig, I would start thinking that maybe I had a plasmid here or an area of horizontal transfer that we needed to look at. So this is what is in that genome report. It's a really important document giving you indications of problems and also giving you statistics on how good you should consider your genome to be. In the next video, we're going to look at your genome integrated into the resource. See you there. Bye.